Shit.
Hi, my name is Yari Rodriguez.
My name is Jack Vang.
My name is Justin Zhang. We're biochemistry students from Fresno State, and today our infographic is on Drosophila melanogaster as a model for biomedical research. Drosophila melanogaster is a common fruit fly with a very rapid life cycle, which makes it an ideal choice as a model organism. The fruit flies are easy to maintain and keep alive and are very susceptible to various tests performed in a biomedical research. In a recent study done, Drosophila melanogaster was used as a model for testing additives. Extracts found in brown algae was used as a supplement in the fruit fly's diet and was found to increase its lifespan. Through the uses of various laboratory techniques, these effects on Drosophila can be observed.
Much of the techniques that were used for the experiment on the fruit fly were also lab techniques that were used in our Chemistry 156 lab. These techniques included things like spectrophotometry, transformation techniques, purification techniques, gravimetric analysis, and lysis. But the ones that we will be focusing on for these purposes are liquid chromatography, the proper use of micropipettes, lysis, and spectrophotometry. For liquid chromatography, this technique purifies compounds based on the polarity and the hydrophobicity of particles. It's important that lab students are able to use micropipettes correctly in order to efficiently execute experiments. So this includes transferring solutions from one container to another using a forward pipetting method. For the technique lysis, this includes breaking down cells without damaging sensitive proteins and DNA. The last technique, spectrophotometry, measures how much a chemical substance absorbs light.
In the aforementioned lab, Drosophila melanogaster was used to test brown algae extract. A similar experiment would be to use extracts found in microalgae. Lipids in the form of oils can be extracted from green microalgae and then screened with the fruit flies to test for any health effects.
Since macroalgae has high carbohydrate content and it can only yield small amounts of fat and oils, we decided to use microalgae because it yields more fat and oil due to its high lipid content. To grow the microalgae, a microalgae culture is inoculated in a photobioreactor containing nitrate and phosphate-rich pistachio water for a cycle of 12 hours of day and 12 hours of light and 12 hours of darkness for a total of 17 days. To remove the lipid extract from the microalgae, we centrifuged the cell culture, extracted the lipid by heating. We then separated the solid from the extract by filtration. The final step is to remove the solvent by vacuuming, leaving us with just the extract. Over on this graph, there are two groups that contain the biomass of the microalgae strain Chlorella sorokiniana. The difference between the two is the change in diurnal cycle in which the microalgae in group 1 has 12 hours of light and 12 hours of dark, and in group 4 which has 16 hours of light and 8 hours of dark. This shows that in a controlled environment, the algae can be stressed with more light to yield more rich oil. When testing the oil extract on Drosophila melanogaster, the life expectancy went up due to the digestion of these oils. We believe a similar effect can be seen on humans since they contain a similar genome to humans.